Hello student, welcome to my YouTube channel Botany Education. Today we will discuss about plasmid. Plasmid are extra chromosomal small circular supercoiled double-stranded DNA molecule present in all species of bacteria. It is very different to main DNA. The term plasmid was first introduced by Joshua Lederberg in 1952. They are distinct from the normal bacteria genome and non-essential for cell survival. It is like the secondary metabolism of the plants, which is not essential for survival, but it is essential for competition or other things. Plasmid are also known as sex factors, conjugates, extra chromosomal replicants. Sex factors are plasmid, thus they are helping in the conjugation process of the sexual reproduction in bacteria. This is the well-labeled diagram of the bacteria with presenting plasmid and nuclear DNA. That's how plasmid is different from the nuclear DNA. And this is another diagram of plasmid and bacterial DNA representation. And this diagram is represent conjugation or sexual reproduction in plasmids. Here plasmids are transferred to the second cell, and the both cells are getting positive or male cells. Here are some properties of plasmid. Plasmid are extra chromosomal DNA element, replicate independently, contain genes that are non-essential but often beneficial to the bacterium. Plasmid size varies from 1 to over 1000 kilobyte. Absence of plasmid doesn't kill bacterium, but their presence provides additional benefit to the bacterium cell. Like secondary metabolism are beneficial to the plants, and plasmid is also show same character as secondary metabolites. Certain plasmids are known to be linear. Example, Borrelia burgdorferi. Copy number and compatibility is a term used in the plasmid. Copy number, it refers to the number of copies of a plasmid present in a cell. Larger plasmid are present in a smaller number, and the small plasmid may be found in high copy number. Okay, then compatibility, it refers to the ability of two different plasmid to coexist stably in a same bacterial cells. And now, as we know, the plasmid is extra chromosomal DNA, then genes of plasmid are present. Plasmid contain both trazine and ORIT site. The effector is self-mobilizable. It can transfer itself to other cells. This transfer initiates at ORIT site. This ORIT site is the origin of replication site. The factor also contain insertion sequence and transpons in TN1000. ORIT site is cut by specific endonuclease, which is known as relaxase endonuclease. Based on the transfer genes, there are two types of plasmid, conjugative and non-conjugative. Conjugative plasmids are those plasmids in which trazine are present, and non-conjugative are those plasmids in which trazine is absent. And on the basis of functional properties, there are 6 main classes of the plasmid: F plasmid or fertility plasmid, R plasmid or resistant plasmid, COL plasmid, degradative plasmid, virulence plasmid or cryptic plasmid. Fertility plasmid are conjugative plasmid in which trazine present. In addition to transgenes, it contains a number of other genetic sequences responsible for incompatibility, replication, and other functions. We are also known that bacteria is responsible for the many diseases. That's our plasmid are those plasmids that carry and transmit antibiotic resistance genes from one bacterium to another are known as resistance. These plasmids provide protection on the bacteria both from human medicines and from antibiotics produced naturally in the soil. COL plasmid. These plasmids synthesize protein colicins and also known as bacteriocinogenetic plasmid because they produce bacteriocins. Degradative plasmid. These plasmids are the group of plasmids which enable to the digest of unusual substances like toluene, xylene, naphthalene, camphor, etc. Virulence plasmid or tumor-inducing plasmid. Plasmids carry genes for various characters which help bacteria to infect higher organisms, both plants and animals, including humans. Bacteria having virulence plasmids are able to attack animal cells by attacking to their cellular membrane and they release toxins. The last one is cryptic plasmid. There are no specific role found of cryptic plasmid. PBr322 plasmid is the example of a plasmid. PBr322 is a purpose-built plasmid vector and was one of the first widely used cloning vector. It is a 4361 base pair double-stranded cloning vector. This plasmid vector has been put together from fragments originating from 3 different naturally occurring plasmids. This is the well-labeled diagram of plasmid PBr322. The nomenclature of PBr322. Why this name is chosen for the PBr. Created in 1977 in a laboratory of Herbert Boyer at the University of California, San Francisco. P indicates as a plasmid. Br indicate Bolivar and Rodriguez, the two researchers who developed it. P22 distinguishes those plasmids from others developed in the same laboratory. 3 PBr322 has two antibiotic resistance genes. First one is AMPR and the second one is TETR. AMP indicate amplicin resistance protein and the TETAR indicate tetracycline resistance protein. Construction of PBr322. PBr322 is not a naturally occurring plasmid. It is manufactured by following steps. First, the AMPR gene originally resides on the plasmid RSF2124 and the TETR is derived from PSC101, the second antibiotic resistant plasmid, and third one is the origin of replication is derived from PMB1, which is closely related to the colicin producing plasmid COLE first. There are some uses of PBr322. It is widely used as a cloning vector. In addition to this, it has been widely used as the model system of study of prokaryotic transcription and the translation. Thanks for watching. Please do subscribe my YouTube channel for the new updates and the new videos.